In the mature flower of Amaryllis, when the anthers burst, pollen grains are liberated. The female part of the flower is the pistil, which consists of the stigma, the style, the ovary, with one or more ovules. Embedded in the ovule is the embryo sac, which contains antipodal cells whose function is unknown, polar nuclei, the egg cell, on either side of which are the synergids, which seem to function as nurse cells for the egg. In nature, pollen grains are transferred to the stigma, where they germinate and grow down the style. Similarly, the tube of a grain germinating in sugar solution increases in length. The cytoplasm, the vegetative nucleus, and the generative cell migrate from the grain into the tube. The vegetative nucleus is slightly ahead of the generative cell. In the sequence which follows, only the generative cell can be seen moving in the cytoplasm. Several hours after the start of germination, the generative cell divides, forming 2 sperm cells. The tube grows very rapidly at rates greater than 500 microns per hour. Growth of the pollen tube is restricted to a zone of about 5 microns at the tip. The pollen tube, like the root hair, grows only at its tip, unlike most plant cells, which grow over their entire surface. During germination and growth of the pollen tube, the cytoplasm streams very actively. When the pollen tube reaches the ovary, it enters the ovule through the micropyle, passes between cells of the ovule, and penetrates the embryo sac, entering one of the synergids. The tip ruptures in the synergid, and the sperm cells are released. When the sperm nuclei are freed from surrounding cytoplasm, one nucleus fuses with the egg nucleus to form the zygote. The other nucleus fuses with the polar nuclei to form the primary endosperm nucleus. Double fertilization is thus completed.